Welcome back to the deep dive. Today, we're looking at something that, honestly, it sounds like it was ripped straight out of a cyberpunk novel. We're talking about engineering a living, breathing army to hunt down cancer.
It does sound like fiction, doesn't it?
Yeah.
But this is very much science fact.
Yeah.
We aren't just talking about a new pill or, a different kind of chemotherapy. This is about rewriting the fundamental kind of immune cells.
To create these mass-produced, off-the-shelf cancer killers.
Exactly.
We're basing this conversation on a really dense, fascinating review paper. It's titled Human Pluripotent Stem Cell-Derived Innate and Adaptive Immune Cells for Cancer Immunotherapy.
It's a mouthful.
It is, but I've read through this, and the sheer ambition of what these scientists are doing is it's mind-blowing. But before we get to the cool super soldier cells, we have to talk about the problem, because right now, the best treatment we have, CAR-therapy, it has a major flaw.
It does. It's a logistical nightmare.
A bottleneck, that's what the source calls it. Walk us through this, because my understanding is that currently, if you have cancer, they have to use your own blood to cure you.
That's the current gold standard, yes. It's called autologous therapy. And here's the reality for a patient. You're diagnosed with an aggressive lymphoma or leukemia. We have to take blood from you.
Okay.
Then we have to ship that blood to a specialized manufacturing facility, often across the country, sometimes to a different continent.
So your cells are literally on a plane.
Literally.
Yeah.
And while all that shipping and manufacturing is happening, the patient is just waiting.
Just waiting and getting sicker.
Exactly. And that wait time, what the industry calls vein-to-vein time, can be weeks. For a patient with aggressive cancer, 3 or 4 weeks is an eternity. They might get too sick to even receive the treatment by the time it gets back.
The source dropped a statistic that really scared me. It said something like only 25% of eligible patients actually get the treatment.
That's the tragic reality. Between manufacturing delays, the incredibly high cost, and the fact that sometimes the patient's own T cells are just too exhausted from fighting the cancer to be engineered effectively.
So 3/4 of people who could be saved
Aren't.
They miss out.
So the mission of this paper and our deep dive today is to fix that bottleneck. The authors are proposing a huge shift from autologous to allogeneic.
Right. Think of it like a suit. Autologous is a bespoke, custom-tailored suit. It's made from your own fabric, fits you perfectly, but it takes forever to make.
And it might not even be finished in time.
Exactly. Allogeneic is off the shelf. You walk into the store, you pick a high-quality suit off the rack, and you can wear it that same day.
But to make an off-the-shelf cell, we can't just use random donor blood, right? I mean, we'd need an unlimited supply.
Correct. Donor blood is finite. So the solution is to use stem cells, specifically induced pluripotent stem cells, or iPSCs.
This is what you called the infinite clay idea. That's a great analogy. iPSCs are the raw material. We can take a single skin cell, reprogram it into a stem cell, and that stem cell line effectively becomes immortal. We can expand it indefinitely and then, and this is the key part, differentiate it into any immune cell we want.
NK cells, T cells, macrophages.
All of them. We don't need a constant stream of donors anymore. We have a factory.
Okay, I want to mentally walk through this factory because the paper breaks this down with incredible detail. Let's call this segment 1, the manufacturing floor. How do we actually take a skin cell and convince it to become a stem cell? That sounds like reversing time.
It essentially is. The process is called reprogramming. You expose that skin cell to a specific cocktail of proteins called Yamanaka factors.
And these just tell it to go backwards.
They act like a reset button on the cell's hard drive, wiping its memory of being a skin cell and returning it to a pluripotent state, meaning it can become anything.
But the text flagged a safety issue here. Something about viruses.
This is a huge historical hurdle. In the early days, to get those Yamanaka factors inside the cell, scientists used retroviruses. And the problem is, retroviruses work by stitching their own DNA permanently into the cell's genome.
So you're leaving genetic graffiti on the wall.
It's worse than that. It's like throwing a wrench into a delicate machine. If that viral DNA lands in the middle of a tumor suppressor gene, you might accidentally turn your cure into a cancer-causing cell.
The ultimate irony. Okay, so how do they get around that now?
The paper highlights the new standard, transgene-free methods. They use things like the Sendai virus or episomal vectors. Think of these as Post-it notes. They deliver the instructions, reset yourself, and then they just fall off and disappear. They don't integrate.
No footprint, no accidental cancer risk.
Zero. It's a much safer system.
Okay, so we have our safe blank slate stem cell. Now we need to turn it into blood. And this part of the paper got really weird. It mentioned something called the hemogenic endothelium.
Yes, the HE. This is one of the coolest processes in developmental biology.
To me, it sounded like blood cells popping out of a wall.
That is literally what happens. Imagine a blood vessel. The wall of that vessel is made of endothelial cells. In this specific developmental stage, those flat wall cells start to round up and physically bud off. They detach and float away as brand new blood cells.
That is bizarre. So the walls of the factory literally turn into the product.
Biology is incredibly efficient. But here's the catch, and the paper is very specific about this. There are two waves of this blood-making process. The first one is the primitive wave.
Primitive sounds like something we don't want.
Generally, no, not for this. The primitive wave makes cells that are good for a developing embryo-like macrophages that just sit in tissue. But they aren't the killers we need for cancer. We need the definitive wave. That's the one that creates the real soldiers. The T cells, B cells, and NK cells.
And the paper mentioned a specific chemical signal to ensure we catch that second, that definitive wave, SB something.
SB431542. It's a mouthful.
It sounds like a license plate. Why is that chemical so important? What does it do?
It acts like a railway switch. In the cell, there's a signaling pathway called active and nodal. If that pathway is open, the cells tend to stay primitive. SB431542 physically blocks that pathway. It forces the cells to ignore the first exit and keep going down the track toward the definitive lineage.
So by adding this chemical, we are telling the factory, don't make janitors, make soldiers.
Precisely. We use that, combined with other signals like want beta-catenin, to force the production of the immune warriors we actually want.
Okay, so just a quick recap of the factory floor. We take a donor cell, use a safe post-it note virus to reset it, then we guide it to become a blood vessel wall, the hemogenic endothelium.
Yep.
And then we use that chemical switch, SB431542, to force it to pop out the definitive soldier cells.
You've got it. The factory is running. Now we have to decide which soldiers to build.
And this brings us to segment 2. The innate warriors. The paper spends a lot of time on natural killer cells, or NK cells. Why start with them? Why not the famous T cells?
Safety again. Remember, we are putting someone else's cells into a patient. If you put my T cells into your body, my T cells will look at your healthy organs, see them as foreign, and attack. That's called graft versus host disease, or GVHD. It can be fatal.
But NK cells don't do that.
NK cells are much safer. They don't attack the host in the same way. But there is a flip side. Your body might attack them. That's host versus graft.
Okay, so they're safe for us, but maybe a bit fragile. How do they actually kill the cancer? The paper talked about a missing self hypothesis.
This is a brilliant evolutionary mechanism. Think of a T cell like a security guard looking for a specific face on a most wanted list. Cancer cells know this, so they often just take their picture off the wall. They stop displaying their ID card, their MHC class I marker.
So the cancer goes invisible to the T cell guards?
Exactly. But NK cells work differently. They aren't looking for a specific face. They're just checking that everyone has an ID card. If an NK cell bumps into a cell and says, show me your ID, and that cell has no ID, no MHC class I, the NK cell kills it immediately.
It assumes anything hiding its identity is a threat. That is a perfect backup system.
It is. What the T cell misses, the NK cell catches. But we aren't satisfied with natural NK cells. The paper details how we genetically engineer them to be super soldiers.
Let's talk about those upgrades. The first one was CD16A.
Right. CD16A is the hand that NK cells use to grab onto antibodies that are coating a tumor. But in nature, that hand is kind of weak. It can break off, so scientists engineered a high-affinity, non-cleavable version.
So a bionic hand that never lets go.
And never breaks. It massively boosts the cell's killing power. Then they take the parking brake off.
That's the CIS gene, right?
Yes. CISH is a gene that naturally limits the NK cell's activities to prevent it from burning out. By using CRISPR to knock that gene out, we create an NK cell that is hypermetabolic. It has more energy, it's more aggressive, and it lasts longer in a fight.
But wait, go back to the rejection issue. You said the patient's body might still attack these foreign cells. The paper described a stealth strategy that just sounded incredible. A triple edit.
Oh, this is pure biological camouflage. I love this part. Edit 1. They knock out a gene called B2M. This removes that main ID card, the MHC class I marker, from the cell surface.
So the cell becomes invisible to the patient's T cells?
Right. But remember what we just said. If you have no ID, who attacks you?
The patient's NK cells. They'll see the missing self.
Exactly. So that leads to edit 2. They force the cell to overexpress a molecule called HLA-E. HLA-E is a decoy. It's a fake ID that specifically tells the patient's NK cells, nothing to see here, move along.
That is so sneaky. And the third edit.
Retaining HLA-C. It's another layer of protection, a backup disguise in case the HLA-E trick doesn't work perfectly on every single one of the patient's NK cells.
So we have an invisible assassin holding a fake ID with a backup disguise. That's incredible. Now, briefly, before we leave the innate system, what about macrophages? The paper calls them CAR-IMs. Why do we need them if we have these super NK cells?
Because some tumors are like fortresses. Solid tumors, pancreatic cancer, brain tumors. They have this dense physical structure. T cells and NK cells often just get stuck at the gates. They can't get in.
But macrophages can.
Macrophages are designed to crawl through tissue. They're the cleanup crew. They can infiltrate that dense fortress. The trick is keeping them on our side.
The M1 versus M2 thing.
Right. Inside a tumor, macrophages often get corrupted into an M2 state where they actually help the tumor grow. We have to engineer them to stay locked in the M1, the tumor-eating state. And the beauty of iPSCs is that we can create billions of these pure M1 macrophages, which you could just never get from a donor.
Okay, let me pause and check the map. So far, we've built the factory. We've deployed the innate warriors, the NK cells with their bionic hands and stealth cloaks, and the macrophages that can storm the castle. But the paper dedicates a huge section to the adaptive immune system. The T cells. You mentioned earlier that regular T cells are dangerous because of GVHD.
They are. The T cell receptor, the TCR, is what causes GVHD. So if we want to use T cells off the shelf, we have to do something pretty drastic.
You have to blind them.
We have to surgically remove their weapon. We use CRISPR to knock out the Tranca locus. That completely deletes their natural TCR. Now the T cell is harmless to the patient.
But if it has no receptor, how does it find the cancer?
We give it a new weapon, a chimeric antigen receptor. That's the C in CAR-T. We blind it to the patient, but we give it a heat-seeking missile for the tumor.
But the paper seemed way more excited about this unconventional squad. The gamma delta T cells and the INP T cells. Why are they getting so much attention?
Because they solve the safety problem naturally. These cells, they sort of sit on the border between innate and adaptive immunity. And crucially, they don't rely on the standard MHC matching system.
So you don't need to CRISPR out their receptors.
Exactly. You can put my gamma delta T cells into you, and they won't attack your healthy organs. They naturally target stress signals that pop up on cancer cells. They are inherently safe.
If they're naturally safe and effective, why haven't we been using them all along? Why bother with the dangerous ones?
It's a numbers game. Take iNKT cells invariant natural killer T cells. They're incredibly potent, but they are so rare they make up maybe 0.1% of your blood.
So you can't just ask a donor to give you a bag of them.
You'd need to drain the blood of 1,000 donors to get one therapeutic dose. It's just impossible. But with the iPSC factory,
you find one iNKT cell.
Well, find one, you reprogram it, you grow it, and suddenly you have a billion clones of that rare superpowered cell. The iPSC technology turns a biological oddity into a mass-producible drug.
That is the aha moment right there. It's democratizing the rare cells. But we have to look at the scoreboard. The paper discusses a real-world trial, FT596, by Fate Therapeutics. This was an iPSC-derived CAR-NK cell. How did it do in people?
It was a mixed bag, which is typical for this kind of pioneering science. The good news, it was incredibly safe. With standard CAR-T, we worry about cytokine release syndrome.
The immune system going nuclear on the patient.
Right. We didn't see that here. No neurotoxicity. And the other good news, it killed cancer. Patients went into remission.
But there's a but.
The but is persistence. The cells just didn't stick around. They went in, fought hard, cleared the tumor, and then they died off. And once they were gone,
the cancer could come back.
In some cases, yes. Or the patient would need more doses. Unlike the autologous cells that can sometimes live in your body for years, these off-the-shelf cells seem to burn out faster.
Before we get to how to fix that, there was one case study that I found absolutely fascinating. It wasn't about cancer at all. A patient with systemic sclerosis.
The QN139B case.
Yeah.
This is huge. Systemic sclerosis is a brutal autoimmune disease where the body attacks itself, causing fibrosis scarring of the skin and organs.
And they used these engineered cells to treat it.
They did. They used an iPSC RRNK to hunt down the patient's own B cells, the ones causing the autoimmune reaction. And it worked. The fibrosis reversed.
That implies we aren't just looking at a cancer cure. We're looking at a reset button for the entire immune system.
Exactly. Imagine curing lupus or rheumatoid arthritis by wiping out the faulty cells with an off-the-shelf engineered hunter. It's a whole new field of medicine.
Okay, we're in the final stretch, segment 4. Grand challenges. We established that persistence is the Achilles heel. The cells die too fast. How do we keep them alive?
We have to give them a packed lunch. The paper suggests engineering the cells to produce their own survival signals, specifically a cytokine called IL-15.
IL-15 is like a battery pack.
Think of it as a constant survival signal. Usually the body provides it, but these foreign cells might not get enough. By engineering them to make their own IL-15, they become self-sustaining.
And what about the tumor fighting back? The tumor microenvironment sounds like a hostile place.
It is. The tumor releases these poison gases, chemical signals like TGF-beta that shut down immune cells. It also puts up stop signs, activating checkpoints like NKG2A on the killer cells.
So the tumor says, stop attacking me, and the immune cell just obeys.
Unless we engineer it not to. We can use CRISPR to delete the TGF-beta receptor. We can delete the NKG2A receptor. It's like giving the soldiers gas masks and cutting the wires to the retreat alarm. They just keep coming.
Now, I want to end on a logistical idea the paper brought up. We talked about how hard it is to make a stealth cell. That triple edit is complicated. But the paper suggests a different route, the haplobank.
This is a smarter, not harder approach. Instead of engineering one cell that's invisible to everyone, why not just find donors who are already super compatible with a lot of people?
Homozygous donors.
Right. If you have a donor who is homozygous for common immune types, common HLA types, their cells will match a huge chunk of the population. The stats in the paper are wild. In the UK, if you find just 10 of these specific super donors,
Just 10 people.
just 10 people, their cells would be a partial match for nearly 40% of the population.
That is staggering efficiency.
And if you expand that to just 150 selected donors, you cover 93% of the population.
So we don't need a magical universal cell. We just need a library of about 150 cell lines.
Exactly. You walk into the hospital, they check your immune type, and they pull vial number 42 from the freezer. It's not a perfect match, but it's close enough. It makes the whole supply chain actually solvable.
But last safety check, the paper did mention one final risk, insertional oncogenesis.
It did. Anytime you insert a gene, like the key AR, there's a tiny risk you interrupt a healthy gene and cause cancer. The advantage of the iPSC factory is quality control. With autologous therapy, you're in a rush. You engineer the cells and put them back at the patient days later. You don't have time to sequence every cell.
But with iPSCs.
You do the engineering on the master stem cell line. Then you can sequence its entire genome. You can check every single base pair to ensure the insertion is safe. Only after that verification do you start the factory. It allows for a level of safety that autologous therapy can simply never match.
So bringing this all together, we are witnessing a massive shift. We are moving from the artisanal, custom-crafted era of cell therapy to the industrial revolution of immune cells.
That's the perfect way to frame it. The science is solid. The safety mechanisms are in place. The only thing left to really solve is durability.
So for everyone listening, here is the question I want you to chew on. Put yourself in the patient's shoes. You have a choice. Option A, you wait 4 weeks for a perfect match therapy made from your own cells, knowing it might fail to manufacture. Or option B, you take an off-the-shelf dose today. It works. But because of that persistence issue, you might have to go back once a month for a top-up dose, like a biological subscription service. Which do you choose?
It's the classic trade-off, precision and delay, or accessibility and maintenance.
Yeah.
But for the first time, we actually have the technology to offer that second choice.
And as we saw with that autoimmune case, this might be the technology that lets us hit reset on a lot more than just cancer. Thanks for taking this deep dive with us. It's a lot to process, but the future looks, well, engineered.
Stay informed.
Stay curious. We'll see you next time.